So good morning, everyone. My name is Mohammad Yaghmour. I'm the Orchard Systems Advisor for Kern County. And, today I'm just going to talk about the horror rot and how to best manage this problem. As we all know, this disease usually takes place we start seeing this problem as we get close to harvest. However, this disease start infecting the trees before harvest. So generally speaking, when we are talking about diseases, okay, there is three major factors that contribute to any disease. Once we learn and get information about what causing the disease, we will have better knowledge on how to combat or treat this disease. The three major factors are pathogen. So we really need to have a good idea on what is causing the disease. In terms of horror rot, I will explain later on that we have several pathogens that can cause this problem. We will need to understand the factors on the almond, which is the host part, what makes the almond more susceptible, whether it's varietal differences, whether the phenology of the hull split and other factors that will make the plant more susceptible to this disease. And lastly, the environmental factors, such as whether it's a temperature, relative humidity, etc., that is causing a disease. So when it comes to horror rot, previously there were two major pathogens that are causing horror rot. The first one is Monalinia species, and mainly it's the Monalinia fracticula, which is also causing the brown rot in almonds. The other one is Rhizopus stolonifer, which we see more down here in the valley. And in the past few years, okay, we have seen Aspergillus niger as causing more and more problems, causing horror rot here, especially in Kern County and the southern part of the San Joaquin Valley. So, which I've been working on for the past two years. So now those are the three major ones. All right, and I will focus mainly today on the Rhizopus, and I will give an update on the Aspergillus niger part. So what is horror rot? From its name, horror rot starts with the infection of the hull. If you see my cursor here, you can tell, that this nut compared to the other healthy ones start to rot from the inside, and you can see it on this one. What it will cause later on as the fungus is growing between the hull and the shell, the fungus, for example, Rhizopus stolonifer, will start producing a pathogenicity factor. In this case is the fumaric acid. The fumaric acid will start moving into the vascular system, and we'll start seeing shriveling of the leaves. So you can see here how the leaves are shriveled compared to healthy lush leaf on the left here. So as you know, the disease progress, sometimes we can see the whole branch as the fumaric acid moves into the branch, it will kill the branch. So it's worth noting that horror rot is not going to affect current year's yield. But what it's actually doing and why we care about horror rot is that, horror rot is killing the spurs and killing the shoots that are bearing next year's yield. So under severe conditions, we are expecting to see yield reductions in the next year, and so. So moving on, I'm going to talk about the signs. How can we tell which pathogen is causing the horror rot in my orchard? So for Monalinia species, usually we find it in the northern part of the of the state, especially in the Sac Valley, where we have more rain compared to the San Joaquin Valley, and especially compared to the southern part of this valley. So in Kern County, I'll be honest, I didn't see it yet, and I don't expect to see it. It's more of a dry region. So if you look at the lesion here, you will see it's the tan, brown to tan spores on the outside of the lesion. Here they are. So if you see that, that will tell you that you are dealing with the brown rot. All right, and to deal with the brown rot, it's a different way to combat hull rot, as I mentioned later in my talk, compared to rhizopus. So what are the sources of inoculum? The sources of inoculum for brown rot is usually infected almond and stone fruit twigs. So any twigs infected from past seasons, or the mummies that stay on the tree that were infected with brown rot, okay, can be a source of inoculum. So this will tell us later on, when we are going to deal with brown rot, removing the mummies and sanitation is not only for navel orangeworm. I don't want to get ahead of myself here, but it can always help if they are a source of inoculum for other diseases. That will always going to help us combating those diseases too, as well. So moving on to the second causal agent, all right, which is rhizopus. Rhizopus, as I mentioned earlier, it produces fumaric acid, and it's always fuzzy. If you look here, this is close up. When it grows between the hull and shell, okay, it looks black spores, it's fuzzy, but the color is not as black as the Aspergillus niger. So it's between gray to black in color. When it's producing the fumaric acid, all right, and you come and remove the bark, as you can see here, you will see a black streaking, and that is due to the movement of the fumaric acid and killing the shoot. You can see that, in this twig, one of those two fruits got the rhizopus, and the fumaric acid was moving in that direction all the way, and it reached to this point, right? So the sources of inoculum here for this pathogen is totally different compared to monalinia. And the source of inoculum here is the soil. So knowing that, we know that, those spores are going to get on the fruit surface, most likely by causing dust. So they have to move on the fruit by the soil particles movement, which is usually the dust. So the third pathogen here is Aspergillus niger, okay? And Aspergillus niger spores look exactly jet. They are flat. They are not fuzzy like the rhizopus. This is one. And they usually, you will see it between the hull and the shell. That's another one. And they are really dark black compared to the rhizopus. And they look like a powder. So if you open the fruit, that's what you are going to see. And it's important also to know sometimes you will have both the rhizopus and the aspergillus on the same fruit. Previous work had showed that there is varietal differences, and I was talking to earlier today to Brent Halls about this data. This data, David Doll and Brent Halls published it on the Almond Doctor website, showing the differences between the different varieties in terms of their susceptibility to hull rot. It's well known that the nonpareil is very susceptible to hot rot, and that's what I mainly work on here. When I look at hot rot, I mainly look at the nonpareil and record the data for it. And pretty much, hot rot in many orchards, it's 50% of the orchard. So that brings the vulnerability of those orchards to you have 50% of your orchard that might be vulnerable to this disease. In Kern County, the least susceptible one is Monterey. And we have quite a bit here of orchards here in Kern County that are 50% nonpareil and 50% Monterey as a pollinizer. So it's important to know which varieties are the most susceptible and try to manage based on what we have in the orchard. The other important part when we look at what are the factors that are contributing to this disease, the most important also factor is what is the most susceptible fruit stage to those pathogens. So there has been quite a bit of work that was done by Dr. Jim Atscavish that is showing that the B2 stage, when we are seeing a slight separation here in the suture, that this is the most susceptible stage. So why it's important to know the most susceptible stage, because that is later on the stage that I would like, or the grower would like to protect, all right, from the infection, for example, using chemical control. So to apply the chemical control at that point will prevent, the start of the infection, because this is the most susceptible stage. When it comes to Aspergillus niger, in the previous years, I have done similar work looking at 3 major fruit stages, whether the fruit is unsplit or at the B2 stage, which is the deep V, all right, or when the fruit is split less than 1 centimeter, which is pretty much this stage C, which is split less than 3/8 of an inch. So my work, what I did exactly very simple. I have a suspension of Aspergillus niger and come and inoculate by placing. Here is the inoculum being placed on the suture, and later on I come and see what is percentage of spurs showed symptoms. So what we are seeing that close to 40% in both years showed that if we inoculate the fruit at this B2 stage, 40% of the spurs will develop the disease. However, if we inoculate the fruit at this stage, which is the split at C stage, right, close to 80, between 70 and 80% of the spurs will get the disease. So it's important. There's 2 things important. 40% is quite a bit, but the most susceptible stage is actually when the fruit is less than 1 centimeter or 3/8 of the inch open. It's completely open. Also, since we know that the soil is a source of inoculum for both Aspergillus niger and the Rhizopus, this is just to show you what we did. We were looking at the population of those 2 fungi on the fruit surface, and you can tell the population in 2018 start increasing significantly for Aspergillus after hull split, which corresponds exactly with the fruit starting to open after the B2 stage. The same thing happened with the Rhizopus, and here is also the data from 2019. You can tell, as the fruit senesce and we start having the suture open more, all right, getting close to harvest, you start seeing the increase in population of both pathogens. Here is just to show you how much dust there is on 10 fruits. So pretty much what we did is getting those fruits, putting them in a container, plastic bottle, put sterile water on them, and then we plate them. And that's what exactly we get on a plate. So there's quite a bit of dust, and that's how the pathogens get inside the sutures or on the surface of the fruit. So moving on, beside now we knowing how those pathogens move and get to the fruit and what are the sources of inoculum. The other factors that contribute to this disease here is nitrogen. So the work that was done by Sebastian Sa, who's now at the Almond board, had showed significant increase of the incidence of heart rot as we increase nitrogen fertilization. In a nutshell, if we use the nitrogen excessively, right, and we over fertilize with nitrogen, that is a major contributor to heart rot. In previous work, also some of the other factors that may contribute indirectly to heart rot. During the work that was done by Beth Tovedale and David Goldhammer at the University of California, they actually showed that, if you use deficit irrigation, it will significantly actually reduce the heart rot. So if you keep your irrigation as is, probably you will have heart rot due to the other factors. Let's say if you over fertilize with nitrogen, or if you have high inoculum in the soil, you might have a high incidence of heart rot. However, if you use deficit irrigation, they have shown that it will significantly reduce the incidence of this disease inside the orchard. Here you can take a look at, if you are using 100% of the ET, which they use it as a control, they approximately got it 20 clusters that showed this disease. However, when they use the deficit or the regulated deficit irrigation, whether it's at 70% or 85, it significantly reduced it from 20 to 6 and 5 respectively. So that work actually set the steps how we can actually manage the heart rot. So now we know how those different factors, whether it's nitrogen, deficit irrigation, soil, the inoculum in the soil, the varietal susceptibility. Now we know all those informations, right? How they contribute to disease development or reduce disease development. Now we can come to the conclusion that we can use all of those factors to design an integrated heart rot management. So heart rot management, when I say integrated, that's not only using the chemical part of it, but actually using a combination of cultural and chemical control methods to manage this disease inside the orchard. So when we are talking about cultural practices, I mainly will be talking about using the strategic or regulated deficit irrigation. And then I will mention how to use the nitrogen management to manage this disease. And I will also talk about different fungicides that we can use available for us here to manage this disease. And recently, Dr. Azkavij had worked about, did some work regarding the alkaline fertilizers and showed clearly that they can significantly reduce the incidence of heart rot inside the orchard. So moving on to the deficit irrigation, pretty much deficit irrigation is used for mainly there is for its benefits, correct? So, but let me remind you that here we are talking about mild to moderate stress, and we are not going to apply any of high stress on the trees, as high stress on the trees will negatively affect the yields and compromise the trees. So why do we use deficit irrigation? First of all, it will increase hull split uniformity, and that's something we will need. It will help with harvest, as well as it will help with the chemical control, whether it's for navel orange worm or for hull rot. Secondly, the previous work had showed clearly that it reduced hull rot, and this is one of the main reasons why we are using deficit irrigation. So how do we start applying the deficit irrigation? So if you're gonna use it for the first time, it's always recommended to slightly reduce your irrigation by 10 to 20% of the amount of water you apply to the orchard. It's very important that we maintain the irrigation frequency. So if we are irrigating twice a week, we keep the twice irrigation times a week. However, we only reduce the amount of water we add in every time by 10 to 20%. So when you start that and your trees reach 2 to 3 bars below baseline, then you resume normal irrigation until hull split starts. So the whole idea, we want the deficit irrigation to be practiced inside the orchard for 2 weeks once the hull split starts. So once the hull split, we have approximately 1%, then we want for 2 weeks the stress or the mild stress on the trees to be between -14 to -18 bars. You will look at some of the literature and they will choose -14 to -16 bars, which is fine. So you maintain that deficit irrigation for 2 weeks, and after that you return to normal irrigation, which is a full ET, until you start the harvest dry down in preparation for harvest. Now, there is more than one factor that goes in when you are applying the deficit irrigation. It's always important to remember that what we are trying to do when we are starting to cut off the water by 10 to 20% here, right? We want to the tree get into stress, correct? We want the tree to be close to -14 bars, between 14 and 18 when the hull split starts. So that will depend really on the soil type. So you can reach the -14, between -14 to -18 very fast if you have a sandy soil. However, if you have clay loam soil where you have more of a water holding capacity, probably the grower will need to start that process earlier. So usually you start maybe 2 weeks before the normal start. So we are talking here about the hull split starting the beginning of July. Under normal sandy, let's say sandy loam soil, you may start reducing the irrigation water by 10 to 20% by mid of June. However, if you have clay loam soil, maybe you need to start earlier than that, 2 weeks or a week earlier. The whole idea, we want the tree to consume the amount of water that is reserved around the root system. So the soil type here matters. The other thing is, if you are doing it for the first time, or not the first time, also you are doing it regularly, it's also good to use the pressure chamber. If you use the pressure chamber, you know exactly how much stress you are putting on those trees. Because remember, we are only trying to apply mild to moderate stress, and we don't want the trees to be in too much stress during that period, during the hull split. Otherwise, it will adversely affect the tree, and it may have consequences on the yield as well. The other thing is to take in consideration is drip versus micro or flood irrigation if you are using flood irrigation. Remember, it's easier to reach that point with a drip compared to micro sprinkler, as micro sprinkler will put more amount of water on the orchard floor. So those are three things to take in consideration when you are applying the deficit irrigation. There is an important information and good information. You can find it about deficit irrigation and how to apply it. It's a UCNR publication 8515, and I will pass that publication to Ashley so she can include it later on with their distribution as a handout. So it has really good information about hot rot and strategic deficit irrigation. So I'll move on to nitrogen management. So as I mentioned earlier, that the previous research shows that excessive nitrogen will increase susceptibility to hot rot. So it's always very important to follow the nitrogen management plan based on yield and not to over fertilize. All right. And we can always manage the nitrogen fertilization by always also looking at the mid-summer leaf nitrogen being mid-July leaf analysis being within the critical value. It has been reported before that, sometimes you still have your leaf showing normal nitrogen analysis within between 2.2 and 2.5%. However, you still sometimes can get hot rot. So not always the leaf analysis value will correlate with hot rot, because sometimes there's other factors going into that beside the nitrogen. So before harvest, right, it's important to know that we should not apply nitrogen after the completion of kernel fill, as previous research have shown that, adding nitrogen after that part, after the completion of kernel fill and kernel development, will mostly go to the hull, concentrated in the hull, and it can exacerbate the problem. So always follow the nitrogen management plan based on the yield and try not to over fertilize. When we are talking, I'm now going to switch gears and talking about chemical control. So when we are talking about chemical control, here, most of this work has been done by Dr. Mohammad Scavage, and you can find this table here at the UCIPM website. It's pretty much showing the different fungicides and their efficacy here. It's important to remember that none of those chemicals have an excellent and consistent rating when it comes to managing hot rot. However, there are many chemicals that belong to the FRAC group 3, 11, and 19 that provide good and reliable efficacy. Also, Dr. Mohammad Scavage had shown in the past few years that the use of alkaline fertilizers such as dicap and dipotassium, the dicap and cynetis, right, can actually significantly reduce the hot rot incidence inside the orchard and is as comparable as using in terms of their efficacy, they are comparable to the fungicides. The theory behind why they might be effective. So Dr. Mohammad Scavage thinks that, Those alkaline fertilizers actually might be interfering with the fumaric acid. So they are neutralizing the fumaric acid that I mentioned earlier, that it moves inside the shoot and kills the tissue. The timing is always very important when we are dealing with hot rot, right? I have shown earlier the work of Dr. Jemad Skavish that, when it comes to Rhizopus stolonifer, the best time is to apply it when we have the hull split at the percentage between 1 and 5%. And that's exactly when the trees are approximately at the B2 stage. So we are trying to protect that stage from getting infected with Rhizopus stolonifer. In terms of hot rot that is caused by Monalinia fracticula or Monalinia species, so the timing is important here, and it's best managed with fungicide application 3 to 4 weeks before hull split. So, for example, here in Kern County, we start seeing the hull split the first week, sometimes the end of June or the first week of July. That's why here I mentioned in my presentation early June, 3 to 4 weeks before your application for Rhizopus. That's when you apply to manage hot rot caused by Monalinia species. For Aspergillus, all right, Dr. Demis Michelidis did some in vitro work looking at different fungicides in their efficacy against Aspergillus niger. So here is the growth of Aspergillus niger in vitro in a plate that is amended with 10 ppm of each of those fungicides. And we can see here that group number 3, 711, such as Luna Sensation and Miravon, and group number 3, Quash and Tibuconazole, did very well in vitro. Tilt, which is number 3. However, group number A bound is, I believe, it's 11. So A-bound did not do very well, and that prompted me to look at an experiment in the orchard, okay, using A-bound, Quash, and Miravon, which is 711 and 3, that showed good efficacy, and A-bound. While there was no significant differences statistically between the control and the different treatments here, however, we have seen that they did reduce the incidence of hot rot inside the orchard caused by both Aspergillus and Rhizopus. This work has been done only for 1 year. I'm planning to do it, to repeat it again and see what we get in that terms. Hopefully, we're still seeing the same trends and the same results. To manage, actually, this way we can manage both hot rot caused by Aspergillus and Rhizopus at the same time. In another lab work experiments, here I wanted to see the effect of those fungicides, those 3 fungicides on inoculated fruits at the 2 stages. Remember, I mentioned that there's approximately, when it comes to Aspergillus, there is between 40 and 45%. When we inoculate them at the deep V stage, they will 40% of the spurs will develop those symptoms, and approximately 70 to 80% will develop when we have the fruits split at less than at stage C, which is less than 3/8 of an inch. So with this experiment that was done by bottle spray, all right, which really does not resemble what commercially is being done. So we have seen the trends when we use it at the deep V. If we use the chemicals at deep V and then we we inoculate and then we use it, we see the same trend. However, at the split C stage, we had quite a bit of infection with rhizopus. So that is still work in progress. So we will see this year we're gonna repeat this work again and see. Hopefully, we are seeing the same trends using this chemical at the DV and even maybe at the when the fruits are totally split, maybe we can reduce the Aspergillus incidence in the orchard. So when we are looking, I will finish my talk by putting this whole picture. It's very important to understand which agent is causing the hot rot inside the orchard. Based on that, timing of the application for chemicals, whether the use of other cultural practices will help or not, depends on knowing which pathogen we are dealing with. Also, knowing the sources of inoculum is important. So if the soil is for example, Aspergillus and Rhizopus, then dust management should be implemented to reduce the incidence of those spores getting on the surface of the fruit. Varietal differences is also important. If you have more than one variety, all right, that is susceptible to this disease, for example, for Aspergillus, and we know that the best time to apply it is when at the B2 stage, maybe you need to do different sprays. If you spray the whole orchard based on the nonpareil and the other variety is not yet at that stage, the other variety may not get the benefits of that spray. Nitrogen management is important. It's we should not over fertilize. Irrigation management, I have shown that in terms of rhizopus, really deficit irrigation will significantly reduce hot rot incidence in the orchard. With that, I reached to the end of my presentation, and hopefully I have some time for questions. If you have any questions, please go ahead.